Otherwise I have to come down by 42. Okay. Hello, it's a great pleasure to start the conference today, and thank you so much. I'm excited to tell you about a plant that associates with insects, Acacia drepanolobium in Africa. And before I start, I just want to introduce the people who have done most of the work, 4 very talented graduate students and 3 undergraduates, and particularly Dino Martins, my student from who comes from Kenya, and first told me about ant plants and got me interested in the system. So of course, ant plant symbioses will be familiar to most of you. They occur worldwide. They're restricted to the tropics. There are hundreds of species that are myrmecophitic, and of those that make Demacia, there are at least 158 independent evolutions of the Damation-making habit in 50 families of plants, so very widespread. For the ants that live in those plants, some of the ants come and go out of the houses that the plants make for them, but there are over 100 species that are phytoecious. That means that they are obligately associated with plants. They can't be free living without the plant, which makes it a very interesting association in terms of looking at the costs and benefits of this mutualism. And of course, it is a classic canonical example of mutualism. Many of us were influenced by the work of Dan Jansen in the 60s, who really inspired us to go on and study the evolution of cooperation using ant plants as a model system. If you go to East Africa, the Rift Valley, and we make it to Kenya, especially the black cotton soils, these are clayey vertisols in East Africa. They are dominated by a single plant, which is the acacia, the Whistling Thorn Acacia, Vachellia drepanolobium. So it's the dominant tree as far as the eye can see. It's overdispersed. Each tree is effectively an ant colony, and so they don't tend to touch each other. So it makes for a very interesting landscape. It's a classic mutualism of exchanging housing and food for defense of the plant by the patrolling ants. And interestingly, there are commonly at least 3 phytoecious ants in the system. Sometimes there's a 4th species that is not phytoecious, and I won't talk about it today. It's a Chromatogaster shastadi, but the 3 common ones are Mimosi, Nigroseps, and Tetrapona pensigi. When they take over a tree, they tend to take the entire tree. So a single tree would be a single colony, although a colony, if it grows very large, might occupy 5 or 6 trees that are adjacent to each other. So they make a mosaic through this landscape of plants. Many people have worked on this system, especially classic work by Hawking in the 70s, and then the group from UC Davis, Truman Young, Stanton, Todd Palmer, later Rob Pringle, and others have looked at the patrolling behavior and defense and sort of competitive and colonization behaviors of the ants, and how they manage to coexist on effectively the same resource. So we stood on the shoulders of giants by starting to study this system, and this has been very helpful in asking questions. When Dino started his thesis, he was interested in looking at how effective the ants were against large grazers like giraffe and rhino and so on. And as you can see in this very simple but telling graph, these giraffe will approach trees containing all 3 ants equally, but they stay away from the tree containing Chromatogaster mimosi, which is the most bellicose ant. if you wonder what a small ant can keep an elephant or a giraffe from feeding on it, here is what they look like. You can imagine what a nose full of those biting ants might be like, or if they get too close to your eyes or your ears. So they really are very effective at protecting the plant. By the time we started studying this system, it seemed clear that the classic two species mutualism wasn't exactly accurate, that actually these mutualisms consist of a multi-party complex network of interactions. And it was this nature of the third party interaction that interested me, and I especially was interested in myrmecophiles. These are insects that live in association with ants. You can imagine my delight at opening that dimension and seeing the two lycaenid larvae inside. These are ant-tended caterpillars of blue butterflies. They secrete compounds that fool the ants and sometimes reward them. And they effectively parasitize the ant acacias. So over a 5-year period, we observed at least 25 species of lycaenids that parasitize this very rich island resource of trees with ant nests hanging in their boughs. We thought it'd be interesting to do a systematic survey of what was out there in the trees. Right from the beginning, there were many interesting things: flies and fly larvae and beetles and many different Lepidoptera. We collected whole trees and took them apart in the field station. And it was a very easy matter to harvest what was inside. In the old days, there would have no chance of knowing what these things were. But of course, now we can take the larval and juvenile forms, even the eggs, and sequence them and have a pretty good chance of figuring out, at least to genus level, what was inside the domation. So this gives you an example of the results from 22,000 domatia, showing at least 82 different OTUs, or operational taxonomic units, which we call species in this case, of different myrmecophiles. So a very rich system filled with insects. And one of the things we noticed right away, sampling across 2 years or in two very different locations, the site in Kinangela and Suyen are about 200 kilometers apart from each other. But at the two sites and in the 2 years, Chromatogaster mimosi, the most bellicose ant, was also the one that had the most number of myrmecophiles in its colonies. So it may be a fighter against the big grazers, but perhaps that's why the myrmecophiles like it so much, because it is very good at protecting the nest. We found that the ants had very specialized communities inside the nests, and here you can just see one of these correspondence plots of how that sorted out for the different species. And so we found many specialists. There were a few generalists too, of course, but the specialization was highly significant, and we've worked out quite a few interesting life histories. Now, it wasn't just the myrmecophiles that we were interested in. We were also interested in fungi and culturing out the different fungi that were in the domatia and trying to figure out what they were doing. And part of this was because Dino, when he was culturing the system in Kenya, had the ants in artificial glass nests and noticed that the tetrapona had started to cultivate fungus along the side of the glass test tube and seemed to be eating the fungus. So he brought this to my attention, and then he and Chris Baker got together and showed, sure enough, yes, the workers of the tetrapona were harvesting the fungus, whereas the Chromatogaster were somewhat indifferent to it. We then looked more closely with sequencing and collected domatia from all three trees bearing all three ant species at both Kitengela and Impala in Suyen, as I say, or in Laikipia, next door to Suyen. So 200 kilometers different. We also had the greenhouse samples from Cambridge, Massachusetts, so very, very different. And when we sequenced them, you could see that right away by eye here, just looking at a class level, they were very, very different. The tetrapona from the nigroseps there, or the greenhouse samples from the leaves, very different. And another way to look at them with one of these non-metric multi-dimensional scaling plots shows in sort of floating space how really distinctive the fungal communities were in the different in the trees occupied by the different ants. So on the left, including the leaves and the greenhouse samples, and on the right, the trees with the different ant species. And here you can see one of these nice spinning models with the Bray-Curtis distances of the with the fungi on the right and the bacteria on the left. The fungi also have the leaves there, but these were taken from the same domatia. So the bacterial communities are diverse as well. Exactly what they're doing, we don't know. We assume that they may use some bacteria to manage the fungus, but all of this remains to be worked out. Also, how these things are assembled, because it could be the ants have different foraging patterns, very different feeding patterns. That would be one place. But also inside the nest, they have very different sort of extended phenotypes in the nest itself. So Mimosae makes a carton nest, a bit like a wasp, and Nigroseps has little leaflets, and Tetrapona pensigi, you can see the hyphae there already, but it has a sort of popcorn inside the nest. So very different substrates for the fungus to grow. But I was also very interested in how the fungus got there in the first place, because if you open an unopened domatia, it's pristine inside. There are no bacteria or fungi to culture out. So when the queen goes inside the domatium from the get-go, there's a good chance for her to inoculate that fungus, inoculate that domatium with her fungal cocktail. And how might she do that? Well, we know that that queens of Atta, when they found a fungus garden, carry fungal hyphae in the infrabuccal pocket, which they check up as soon as they started the nest to start their fungal cultures. So we took the alates of two of the species, Nigroseps and pensigi, and sectioned them. And these are beautiful sections that Johan Billen did in collaboration. And you can see inside those infrabuccal pockets, there are many fungal hyphae. And indeed, when we sequenced them, we found that we could recapture the diversity to about 80% for the Tetrapona or pensigi, and close to between 90 and 100% for the Nigroseps. So yes, certainly it's a potential source of the fungal community that the queen might be able to inoculate the domatia. And why is this interesting? Well, it's interesting to me because it's possible then that the true mutualists in this system could be between the plant and the fungus if we knew more about what the fungi were doing. And the ants might simply be the vector to carry that fungal cocktail between trees, because after all, it's otherwise difficult to spread your spores in the right combinations across distance of trees. So we got very interested in this, especially with pensigi, because it's not a very good, you sort of wonder, how is this one an associate? Because it doesn't seem to protect the tree very much. It hardly ever seems to be out there eating. It doesn't attack herbivores, but maybe it's a very good vector. We built a bush house. We grew a lot of trees. We were ready to inoculate them with our fungal cocktails to see how the plants might grow just with the fungi, without the ants. And then the pandemic started, and all those trees are still out there, sort of waiting for us. But it led us into an unexpected detour. But that unexpected detour has yielded a very interesting result. We brought back colonies of the ants with us from Kenya, and we were also growing trees in the greenhouse here. And we wanted to look at how the how the ants might affect the growth and architecture of their host trees. Now, almost anyone who's studied these ant plants has thought that they must, because when you see them in the field, it's they have a look about them. You can say, oh, this one probably has nigroseps in it. This one probably has mimosae in it. And Mo Stanton had actually written a very beautiful paper where she talked about how nigroseps might influence the architecture and growth pattern of the tree so that it turns the branches upwards to prevent them from touching the neighboring tree. So there's plenty in the literature to suggest that this might happen, but a full experiment across all the taxa hadn't been done. Of course, we found, yes, there was a big difference in the architecture. And here you can see with Pensigi, the Domatia are much smaller, but there are many more of them on the branches. Just walking into the greenhouse, you can see the branches of the trees that have Pensigi in them, and they're extremely distinctive. So yes, we found that was the case. But that was not the most interesting. Almost as a lark, we wondered, it started as a conversation about the pattern of little feet. And we wondered about what would happen if we didn't have feet on the plant itself, but only the smell of the ants. Would it be possible? I mean, I'm very influenced by the fact that plants will respond to methyl jasmonate as signals between plants. But could it be possible that the ants would respond to the smell of the plants would respond to the smell of the ants? And we tested this by doing multiple experiments, but the fifth iteration of this one, we had the ant nest with a mesh top so that the odors could arise with the seedling. And we put it inside the oven bag. That's like a turkey based in oven bag, so it's sort of chemically inert, for an hour and a half every morning for 6 weeks. And then we randomized the positions in the greenhouse so to control for shading, anything like this. We also controlled for colony size by checking the amount of CO2 coming off those colonies to make sure that CO2 couldn't be contributing an added effect. And we did this in the fifth iteration experiment because we almost couldn't believe the results that we kept getting over and over, which was, yes, the plants respond differently to the smells of different ant species. And they respond in a way that is very particularly satisfying. They branch more in response to their symbiotic ants. Of course, branching means that you're going to immediately be producing more extra-floral nectaries, more Domatia. So it's a branching pattern is also very unambiguous to score. And as you can see here, is highly significant from week 5 onwards, a very different growth rate. In addition, the plants produced fewer extra-floral nectaries when they were inhabited, when they cohabited or smelled the ode Tetrapana pensigi, which also interested us because pensigi is the only ant that it's not interested in the extra-floral nectaries. In fact, it destroys them on the plants that it occupies. And we thought it was very striking that the plants were making very significantly fewer extra-floral nectaries when they were within the range of the smell of Tetrapana pensigi. So just to summarize then, these mutualisms are complex networks, and because of that, you have to be very careful about assigning causality to observed effects because there may be multiple parties involved. And in talking about plants, we were so struck by the sensitivity of the plant in responding to partner smells or odors. What are the receptors? Does a plant have a nose? And also, how common is this? If we look at an ant plant, that's a very sensitive co-evolved system. It may be that many plants have this ability, but it's harder to tease apart and really observe it because the system isn't so tightly knit together. So lots of questions it's raised, and I hope you agree. If some of you wonder about whether ants are sentient beings, well, certainly here's another thing to think about as to whether or not they might be able to smell their ant partners. And with that, I'll thank my lab again and thank the organizers of this conference very much. And thank you for listening. Bye.